Hello everyone. In the class, we couldn't look at the PDB data bank, so I'm doing this screen recording now. This is PDB, Protein Data Bank. It's the educational site of the PDB, and here we have the molecule of the month as a hemoglobin. So it starts with some description of why the blood is red and blue, and it also talks about myoglobin. We will look into it. First of all, I think I'm gonna go here. Yes, so the oxygen binds to the heme group is shown in here, and how the hemoglobin changes its conformation. It says allosteric motion of hemoglobin with an oxygen molecule in turquoise in here. So this heme group changes the shape, and also the whole hemoglobin has change in its shape. And here we have some visualization of the binding of oxygen. First of all, here we have green, the iron atom, and this shape in here is the heme group. And when the oxygen as a molecule binds to iron, it is shown here. And we talked about this histidine and significance of it. It forms a hydrogen bond with the bound oxygen. And we can open J as small, which is a visualization for better visualization. It's coming. So here we have the deoxy and oxy states of hemoglobin. First of all, we have the structure. The two alpha chains and two beta chains are here, and in each of them there's a heme group with an iron in the middle. And now this is in the deoxy state, which oxygen is not bound, and here it is in the oxy state. It's not very clear, so we will choose close up on one heme. Yeah. Now we can turn this around. This is the histidine, iron, and the oxygen molecule in here, the oxy state and the oxy state. You can notice that small conformational change in the heme group as well. So it is kind of significant for this. Activity for the activity of hemoglobin. And okay, so now we talked about the sickle cell hemoglobin, and this is the shape of the sickle cell hemoglobin, which is in a fiber shape, and it forms the fibers inside the red blood cells, which prevents their motion in the capillaries. So now we can look at different configurations of hemoglobin. Now here it says nitric oxide can bind to specific cysteine residues in hemoglobin and also to the irons in heme groups as shown in this entry. So we open that one. Here what it opens as protein data bank, and here it says crystal structure of S-nitrosyl human hemoglobin A. So hemoglobin A, which is bound by the nitric oxide in this situation. Method is X-ray diffraction. So these are the exact crystalline shapes of this molecule. So we can look at the structure. Here you can see 4 polypeptides of hemoglobin. And whenever we pick something, it says what it is. Here, for example, this is a water molecule. Here we have hemoglobin protein part of it, and it shows it is the 71st amino acid, which is alanine. And here again alanine, serine 131. So we can get a close-up visualization here. And here we have the heme group. Okay. Yeah, protoporphyrin 9 containing iron in the middle. A close-up visualization of alpha helixes. See here we have the amino acids, and the critical histidine is in here. Yeah, histidine. It shows. Now we come back and we can look at the ligand interaction. Porphyrin. Here. Okay, this is the nitric oxide, which is bound to the structure where oxygen should be binding to it. And the nitric oxide, this is the porphyrin, this is the nitric oxide and histidine. And here we have the dashed lines, which are hydrogen bonding, which it shows between the histidine and nitrogen in here. It's also nitrogen, by the way. NH, this has an H, and this has an NO, nitric oxide. Okay. Then it shows carbon monoxide as a toxic gas. It readily replaces oxygen at the heme group, as seen in here. So we open this one. Structure of human carbon monoxide hemoglobin, which means hemoglobin which is bound to carbon monoxide. Now I go directly to ligand interaction. What do we have here? Yeah, here. Carbon monoxide is in here, and it is an irreversible interaction, as you may remember. Again, the porphyrin ring. Can get a close-up. Carbon monoxide. We can see the histidine's resonance structure in here, the double bond distribution of the double bond, and again, hydrogen bonding. So here we go back. Truncated hemoglobins. We talked about the adult and fetal hemoglobins are different. And in horse, there is a different hemoglobin. So it can have evolutionary variations that will suit the needs of the organism. Artists. Human deoxyhemoglobin. This is okay. It's this animation in the structure without the oxygen and with the oxygen. Do we have carbon dioxide? It has carbon monoxide. Mm. This is myoglobin. Sperm male myoglobin. This was also in the book. It goes on like that. So okay, we can enter this. This has one protein. Hemoglobin had 4, as you may remember. And it's the one protein, and also there's only, obviously there's only one heme group, but it is very, very similar to hemoglobin because we have the heme group in here, we have iron and also histidine in here. So in water molecules, this sulfate ion, it's in the water molecules and it has alpha helixes, which is like one of the subunits of hemoglobin. And we can ligand interaction. The ligand interaction again is very similar to hemoglobin. You can see all the hydrogen bonding. Histidine. Okay. It doesn't have oxygen in this. All the green ones that are shown here are carbon, and this is iron. These are the red ones, usually are oxygen, and the blue ones are nitrogen. By stacking. Does it show explanation? No. Okay, there are lots of other components to this database, but obviously I'm not a protein scientist, so we can just look around. This is the opening page, this summary, and